Think about those beautiful coastal waters, so inviting. But beneath the surface, there's a whole world we don't always see. And
Well,
sometimes, that includes hidden threats that can really mess with our health and, what, the local economy too.
absolutely. Things like tourism, fishing, aquaculture, these are massive industries.
Yeah, I was looking at some figures. The U.S. marine economy, something like $397 billion a year, supporting 2.4 million jobs. That's huge.
It really is. And closer to home for some listeners, in Great Britain, those seaside overnight trips
Hmm.
worth about 4.6 billion back in 2022.
Wow. So keeping these waters healthy is, well, it's vital.
It's absolutely critical. And one of the key things we look for to sort of gauge the potential risks are what we call faecal indicator organisms,
Yeah.
FIOs for short.
FIOs.
Yeah, these aren't necessarily the harmful bugs themselves, but if they're present, it strongly suggests the water could be contaminated.
Contaminated with things that make you sick.
Yeah, things that can cause nasty stomach bugs, eye infections, that kind of thing.
Yeah.
Studies have shown a pretty clear link.
Right.
That's why you have regulations like the EU's Bathing Water Directive. They set strict limits on how many of these FIOs are, well, acceptable in swimming spots.
Okay, so where do these FIOs actually come from? I mean, they don't just magically appear in the sea.
No, unfortunately not. There are several sources. We're talking about rivers and streams flowing into the sea, obviously.
Makes sense.
Then you have discharges from sewage treatment works. People often call them STWs. And a big one, combined sewage overflows, CSOs.
Ah, the CSOs. We hear a lot about those, especially when it rains heavily.
Exactly. Heavy rain overwhelms the system, and untreated sewage gets released. Plus, you've got more sort of diffuse sources like animal waste washing off farmland, for instance. Research has pointed to all these different pathways.
And it sounds like this problem, it might actually get worse.
That's the concern, yeah. Factors like climate change,
Yeah.
potentially more intense rainfall events.
Right.
Combine that with growing populations, more urbanization.
Huh.
It all likely leads to more runoff, more CSO spills.
Meaning potentially more FIOs ending up in our coastal waters.
Precisely.
Okay, so it's not just knowing where they come from. It's also about figuring out where they go and I guess what happens to them once they're actually in the water.
Exactly that.
Yeah.
Understanding how these FIOs move around the transport and how quickly they break down or die off their fate, that's really crucial for managing water quality effectively.
How so? What does that understanding help with?
Well, it helps inform decisions about, say, wastewater treatment upgrades.
Mm.
It lets us build early warning systems to protect public health.
Like beach warnings.
Exactly. And it helps safeguard sensitive industries, like shellfish farming, which can be badly hit by contamination.
Okay, and this brings us to FIO decay modeling. Sounds a bit technical.
It can be, but the basic idea is pretty simple. It's about predicting how quickly these organisms die off once they're in the seawater.
Right. So how long they survive.
Essentially, yes.
Yeah.
And there are generally two broad approaches to modeling this. You've got data-driven models.
Which just look at patterns in the data.
Kind of, yeah. They can be quick for making predictions based on past observations. But then you have the more mechanistic hydro-epidemiological models.
Okay, that's a mouthful.
It is. But they try to simulate the actual physical and biological processes involved. Water movement, sunlight, salinity, how these things actually cause the FIOs to decay. This gives a deeper understanding.
And that's our focus today, making those complex models better.
That's the plan. We're really going to dive into how we can improve the FIO decay part of these models, making those predictions more accurate.
Okay, so what's the real mission here for this deep dive? What are we digging into?
We're aiming to explore some of the shortcomings of the FIO decay models we currently use, and then look at how scientists are developing enhanced versions for more reliable predictions.
And we're focusing on a specific model.
Yes, one developed by Stapleton and colleagues back in 2007. We'll look at how researchers have tried to refine that model, using a real-world case study to test their improvements.
All right, let's get into the limitations then. You mentioned the basic idea of FIO decay is often treated as a first-order process. What does that actually mean in simple terms?
Okay, imagine you have a big population of these FIOs in the water. First-order process basically means the rate at which they die off is proportional to how many are there at that moment.
So the more you have, the faster they die.
Exactly. And as their numbers decrease, the rate of die-off slows down. It's a bit like radioactive decay, if that helps.
Okay, I think I follow.
And we often talk about something called T90. That's simply the time it takes for 90% of the initial population to die off or become inactive.
Ah, so a measure of how quickly they disappear.
Precisely. And T90 is directly linked to that decay rate, K. A faster decay rate means a shorter T90. And crucially, this decay rate, it's not fixed. It's heavily influenced by the environment.
Like, what sort of environmental factors are we talking about?
The big ones are sunlight, specifically UV radiation, which is pretty lethal to these microbes.
Right, sunshine kills germs.
Basically, yeah. Then there's salinity, how salty the water is. And also turbidity, how cloudy or murky the water is.
Turbidity. How does that affect things?
Well, murky water can actually protect the FIOs a bit, shielding them from that harmful UV radiation. So more turbidity might mean slower decay, paradoxically.
Interesting. Okay, and the Stapleton model we mentioned, how does that fit in? What factors does it consider?
So the Stapleton model, which was developed based on studies in the Severn Estuary and Bristol Channel area,
Okay.
it specifically looks at how solar irradiation sunlight and turbidity influence the decay rate, or the T90. It uses a set of equations to calculate T90.
What goes into those equations?
It takes the turbidity level, the amount of sunlight hitting the water at that time, and it relates it to a sort of baseline sunlight level they used in their experiments.
Okay.
It also includes specific constants, KEB values they call them, for different types of FIOs. They provided values for common ones like Enterococci and E. coli. So essentially you feed it turbidity and sunlight data, and it gives you an estimated T90.
Right. Sounds like it covers some important ground, but we said it has limitations. What are the main problems researchers identified?
Well, this recent research really highlighted two key issues. First, the model doesn't do a great job predicting decay rates at the extremes, either in very low light, like darkness, or under very intense sunlight.
How so? What happens in the dark according to the model?
In complete darkness, the Stapleton model actually predicts an infinite T90.
Infinite, meaning they never die off.
Exactly. No decay at all, which, well, contradicts quite a bit of other research. We know from other studies that FIOs do decay in the dark, just, typically more slowly than in sunlight. There are reported dark decay rates.
Okay, so that's a definite issue. What about the other extreme, super bright sunlight?
There, the model has the opposite problem. At very high levels of irradiation, they calculated a specific threshold around 570 watts per square meter. The model predicts a T90 of 0.
0, so they die instantly.
Poof, gone. Which also isn't realistic. While strong sunlight is certainly bad for them, it doesn't cause instant annihilation.
So the model breaks down at both ends of the light spectrum. Did people try to fix that?
Some researchers, like King and colleagues in 2021, put in a sort of temporary workaround. They just imposed limits on the sunlight values the model could actually use, cutting it off at the top and bottom.
Like putting guardrails on it?
Kind of.
No.
But the authors of the study we're looking at point out that this fix, while practical, might compromise accuracy, especially when real sunlight levels actually go beyond those imposed limits.
Right, you're forcing it to ignore real conditions. And what was the second big limitation you mentioned?
Yes. The second major limitation is that the original Stapleton model simply doesn't account for the effect of salinity.
Saltiness. Does that matter?
It seems to, yes.
Oh.
Other research suggests salinity does influence FIO survival and decay, but it wasn't factored into the original Stapleton equations at all.
Okay, so inaccurate at light extremes and ignores salinity. How did the researchers try to address these problems? You mentioned enhanced models.
Yes, they developed two modified versions, which they called ClipStap and RevStap. And an important point is they aimed for relatively simple adjustments.
Why simple?
So the enhanced models could be more easily adopted and used, especially in situations where you might not have vast amounts of complex environmental data. Keep it practical, essentially.
Makes sense. Let's start with ClipStap then. What's the main change there?
ClipStap primarily tackles that no decay in the dark problem. What they did was essentially impose a minimum decay rate, or a maximum T90 value.
How did they decide on that minimum rate?
They looked at the decay rate the original Stapleton model predicted under fairly low light conditions, specifically 30 watts per square meter, and said, okay, even if the light is lower than this, or 0, the decay rate won't drop below this value.
So it's like setting a floor, preventing the decay from stopping completely in the dark.
Exactly. It ensures there's always some baseline level of die-off predicted, even in suboptimal light conditions, which aligns better with observations.
Okay, that sounds like a straightforward fix for the darkness issue. What about RevStap? You said that one was a bit more involved.
Yeah, RevStap revised Stapleton attempts to tackle both major limitations, the issues at extreme light levels and the missing salinity factor.
Right. How did it handle the dark decay differently from ClipStap?
Instead of just clipping the rate, they looked at how the decay rate changed with sunlight in the model's more reliable range, say up to 260 watts per square meter.
All right.
They calculated the slope, the relationship between light and decay rate, and then extrapolated that slope backwards down to zero light.
Okay, so projecting the trend into the dark zone.
Precisely. This allowed them to estimate a non-zero decay rate in the dark based on the model's behavior in moderate light, rather than just setting an arbitrary minimum.
And the salinity part, how did they add that in?
For salinity, they incorporated an additional decay term. They looked at previous research, notably work by Mancini and others, who had developed relationships between salinity and FIO decay. They adapted one of these relationships, adding a simple calculation where an additional decay rate, Ks, is calculated based directly on the salinity concentration.
So the total decay in RevStap is now due to light plus salt.
That's the idea. The overall decay rate, K, becomes the sum of the light-dependent decay, KDI, calculated using a modified Stapleton approach that handles extremes better, and this new salinity-dependent decay, Ks.
So what does the actual RevStap equation look like, broadly speaking?
Broadly, the total decay K equals KDI plus Ks. The Ks part is just a constant times the salinity. The KDI part is derived from the T90 calculation, which itself is modified from Stapleton's original. It still uses turbidity, but the relationship with irradiation is adjusted, especially at low and high levels, to avoid those infinite or zero T90s.
It sounds like they tweaked the light response and bolted on a salinity effect.
That's a pretty good summary, yes. They adjusted the light component to behave more realistically at the extremes, and added that salinity term based on previous work.
Okay. And how did these new models, ClipStap and RevStap, actually perform compared to the original and, compared to what we know happens in reality?
Well, when they compared the dark decay rates predicted by the models to values reported in the scientific literature.
From real experiments?
Yes. Both ClipStap and RevStap showed much better agreement with those literature values than the original Stapleton model, which predicted zero dark decay. The errors were significantly smaller.
So, definite improvement in the dark.
Definitely. And RevStap also addressed the high irradiation issue by preventing that unrealistic instant die-off. The T90 doesn't drop to zero. They also noted that at moderate to high irradiation levels, say above 150 millimeter-R, RevStap's predictions were actually quite similar to the original Stapleton model.
So it fixes the extremes without messing up the middle ground too much.
That seems to be the case, yes.
Okay, so these enhanced models look better on paper, or at least in dealing with those specific flaws. But the real test is how they perform out in the wild, right? In a complex, dynamic, coastal environment.
Absolutely. That's the crucial next step. You need to validate them under real-world conditions where things like salinity and sunlight are constantly changing.
And this is where the case study comes in. You mentioned Swansea Bay.
Exactly. Swansea Bay in the UK was chosen as the test site for this research.
What's special about Swansea Bay? Why there?
It's a really good natural laboratory for this kind of study. It's on the Bristol Channel, which has one of the highest tidal ranges in the world.
Wow, really?
Yeah, huge tide, so it's very dynamic, hydrodynamically. It also receives FIO inputs from multiple sources, 3 main rivers, the Taw, Neath, and Afan, plus sewage treatment works, discharges, and other drains.
So lots of inputs and lots of mixing.
Precisely. And importantly, there was a wealth of high-quality data available from a project called Smart Coast Sustainable Communities, or SCSC.
What kind of data did they have?
Really detailed stuff. Things like FIO concentrations measured at 30-minute intervals at certain locations, along with data on river flows and STW discharge rates. That high-frequency FIO data is quite rare and very valuable for testing models like this.
And it matters to people locally.
Oh, definitely. The Bay has 2 designated bathing water sites, Swansea Beach and Aberfan Beach, which are popular tourist spots. So understanding and predicting water quality there is really important.
Okay, so they have the location and the data. How did they actually set up the computer model for Swansea Bay? What program did they use?
They used a sophisticated hydrodynamic modeling system called Telemac 3D.
Telemac 3D?
Yeah. It simulates how water moves in 3 dimensions, accounting for tides, river inflows, wind, and how things mix together. They created a detailed digital grid, a mesh, representing the bay's shape and depth.
Okay.
They specified things like how rough the seabed is, where the E. coli sources were the rivers and STWs, and the flow rates and E. coli concentrations coming from those sources.
What about salinity and temperature?
Yep. They set initial salinity conditions and boundary conditions for the seawater coming in, used a constant water temperature based on typical conditions, and included a model to handle turbulence and mixing.
And they had to deal with those sunlight limits for the Stapleton and ClipStap models.
Yes, because those models have issues at extreme irradiation, they implemented those upper and lower limits on the sunlight values used within the Telemac simulations when running the original Stapleton and the ClipStap decay modules. They didn't need the lower limit for RevStap, of course.
That sounds incredibly complex, getting all those different pieces to work together
It
accurately.
really is. And one crucial step, before they even started simulating the E. coli, was getting the salinity right.
Why was that so important?
Because the rivers bring in fresh water, which mixes with the salt water. The pattern of salinity affects how the water moves and mixes, and salinity itself affects decay in the RevStap model. So they needed the model to accurately represent the baseline salinity distribution first.
How did they do that?
They ran the Telemac model for a long precursor period, 38 days, actually simulating just the tides and river flows without adding any E. coli yet. This allowed the salinity levels throughout the Bay model to stabilize and reach a realistic state before they introduced the bacteria sources.
As letting the model settle down first.
Exactly. They also had to use very small time steps in the simulation, half a second, to ensure the calculations were stable and accurately captured the rapid changes, especially near the sources and with the fast tidal currents.
Wow. And the model then predicted E. coli where the real samples were taken.
Yes, the model outputted simulated E. coli concentrations at specific points corresponding to the moving boat transects, where the SCC project collected its high-frequency samples, generally around 1 meter below the surface.
Okay, so after all that careful setup, did the underlying hydrodynamic model, the water movement part, actually match up well with reality in Swansea Bay?
Yes, they did validate that part first. They compared the model's predictions for things like water levels against tide gauge measurements and water current speeds against measurements from instruments called ADCPs.
And matched.
They reported good agreement, yeah. Things like root mean square errors and R-squared values, which are statistical measures of accuracy, were well within acceptable ranges for this kind of coastal modeling.
So they had confidence the model was moving the water around correctly.
Reasonably so, yes. They also checked things like salinity profiles, confirming the model showed the water was generally well mixed from top to bottom in the deeper areas, as you'd expect in a high-energy place like the Bristol Channel.
All right, so the stage was set. The key question then, how well did the different FIO decay models, Stapleton, ClipStap, RevStap, actually predict the E. coli levels measured during those boat surveys?
The results were interesting and a bit mixed.
Oh, not a clear winner.
Not entirely. For one specific sampling transect near Aberfan Beach, during the autumn 2012 sampling period, the RevStap model did show improved accuracy compared to the other two.
Okay, so RevStap worked better there then.
Yes.
Uh.
But for the transects near Swansea Beach, and also for Aberfan, during a different sampling period in summer 2011, the original Stapleton and the ClipStap models actually gave slightly better predictions overall, statistically speaking.
Huh, that is surprising. So the model that fixed the known flaws wasn't always the best fit for the overall measured data.
It seems so. It highlights that overall accuracy depends on more than just fixing specific issues like dark decay.
Oh.
The whole system interaction is complex.
But didn't you say RevStap had a clear advantage under very sunny conditions? Did that show up in the Swansea simulations?
Ah, yes, absolutely. That's where a crucial difference emerged. During the summer 2011 sampling period, when solar irradiation levels got really high, peaking above 500 Wilnir Sadens on some days.
Okay, very sunny.
Very sunny. In the simulations using the original Stapleton and the ClipStap models, they observed these dramatic, very rapid drops in the predicted E. coli concentrations at the Swansea transect. Almost down to zero, very quickly.
The instant die-off problem showing up.
Exactly. When they looked at the decay rates being calculated by those models under that intense sunlight, they were becoming extremely high, leading to this unrealistic crash in predicted E. coli levels.
And RevStap.
Importantly, the simulation using the RevStap model did not exhibit these unrealistic sharp drops during those high sunlight periods. Its handling of higher radiation prevented that extreme decay prediction.
So that confirms the fix in RevStap for highlight actually matters in a real-world simulation.
It does. It's worth noting that during the autumn 2012 period, the maximum sunlight levels were much lower, only around 340 millimeter-ins.
Not high enough to trigger the issue.
Apparently not. And in those autumn simulations, none of the three models showed those rapid, unrealistic drops. So the problem was specific to the high radiation conditions encountered in the summer run with the STAP and ClipStap models.
That's a really significant finding, isn't it? It points to a potential failure mode in the order models under specific but realistic sunny conditions.
It certainly suggests caution is needed when applying those models, or models based on them, in environments that experience very strong sunlight.
Okay, what else did the simulations reveal? You mentioned something earlier about daily changes, diurnal variations.
Yes. The researchers noted that the RevStap models seemed to capture some of the observed daily ups and downs in E. coli concentration at the Iberofan transect during that autumn period.
A daily pattern.
Yeah, a sort of cycle over the course of the day. Interestingly, this diurnal pattern wasn't nearly as clear in the predictions for the Swansea Bay transect, regardless of which decay model they used.
So if it was just sunlight causing the daily pattern, you'd expect to see it everywhere, wouldn't you?
You might think so, yeah. The fact that it was more evident at Iberofan suggests it's not just about the sun going up and down.
So what else could be driving those daily fluctuations at Iberofan?
The researchers propose it's likely a combination of three things interacting. First, the significant input of E. coli from the nearby river Afan.
Which flows out near Iberofan Beach.
Exactly. Second, the powerful tidal action moving water back and forth. And third, the FIO decay rates themselves, influenced by sunlight and maybe salinity.
How do those three things interact to create a daily cycle?
Well, their analysis suggests that when the tide goes out, the plume of water from the river Afan, carrying its load of FIOs, can extend further out into the bay and reach the area where the samples were being taken.
Okay, so low tide brings the river pollution closer.
Potentially, yes. Then, as the tide comes back in, the incoming seawater pushes the river plume back towards the coast and also dilutes the E. coli that reached the sampling point. Plus, during this time, decay processes are ongoing. This cycle, driven primarily by the tide exposing the sampling point to the river plume, could repeat roughly twice a day with the semi-diurnal tides leading to those observed fluctuations.
And why wasn't this pattern as obvious at the Swansea transect?
They believe it's down to the source-receptor connection. The main potential source for the Swansea transect is the River Taw. But the sampling points on the Swansea transect are relatively further away from the Taw estuary compared to how close Iberofan is to the Afan.
Further for the pollution to travel.
Exactly. Their model simulations indicated that the travel time for contaminants from the Taw to reach the Swansea transect was significantly longer, maybe around 10 or 11 hours, compared to perhaps 4 hours from the Afan to the Iberofan transect.
Ah, so the timing is off.
Right. That longer travel time means the arrival of the pollution peak from the river might not line up as neatly with the tidal cycle at the sampling location. The signal gets smeared out, more diluted, and the connection between the source fluctuations and the tidal movement at the beach isn't as direct or strong.
Making the daily pattern less pronounced.
That's the hypothesis, yes. It really highlights how site-specific these dynamics can be.
Okay, so pulling this all together, what are the main takeaways? It feels like while RevStap fixed some clear theoretical problems with the Stapleton model, it wasn't necessarily a slam dunk improvement in terms of overall prediction accuracy across the board.
That's a very fair summary. Yes, RevStap demonstrably improved the representation of decay in very dark and very bright conditions, which is important. But it didn't consistently outperform the simpler models in matching the measured concentrations overall.
What does that tell us?
It really underscores the complexity of the whole system. Predicting FIO levels accurately isn't just about perfecting the decay calculation. It also heavily depends on getting the hydrodynamics right, how the water moves, how river plumes mix, how tides influence transport.
So we need improvements on both fronts. Better water movement models and better decay models.
Exactly. And particularly, more work is needed on understanding and modeling the effect of irradiation, the sunlight, on decay.
In what way?
Well, the researchers point out that the mathematical form of the Stapleton model's relationship between light and decay is a bit different from other common approaches. And, as we mentioned, it was originally developed based on experiments at just 2 light levels, 0 and 1 moderate level, 260 Wilhelm.
Not much data to build on.
Relatively limited, yes. So there's a clear need for more experimental work looking at FIO decay across a much wider continuous range of irradiation levels to really nail down that relationship.
And more field data, too, I imagine.
Oh, absolutely. More long-term measurements of bacteria concentrations both near shore, where people swim, and further offshore, are always valuable for rigorously testing and refining these models.
It was interesting how you mentioned the researchers favored simple adjustments for RevStap compared to maybe more complex decay models out there.
Yes, they acknowledge that other models exist, like one by Hipsey and colleagues, which try to account for different wavelengths of light and other factors.
More detailed physics.
Right. Those models could potentially be more accurate if you have all the detailed data they require, light spectra, specific water optical properties, etc. But the RevStap approach aimed for improvement using parameters that are more commonly available, like total irradiation and salinity.
So it's that classic trade-off between potential accuracy and practical applicability.
It often is in environmental modeling, yes.
And that finding about the diurnal variations driven by tides and rivers, not just sunlight, seems quite significant too.
It is. Some earlier work, perhaps looking more at offshore locations, had suggested diurnal FIO cycles might be primarily driven by the day-night difference in sunlight-induced decay.
But close to shore, it's different.
This Swansea Bay study strongly suggests that in these dynamic nearshore bathing waters, the interplay between freshwater inputs carrying FIOs and the tide moving those plumes around is a major, if not dominant, driver of those daily fluctuations. Sunlight still matters for the rate of decay, of course, but the timing seems heavily influenced by the hydrodynamics.
And this would vary a lot from place to place.
Absolutely. The specific timing and strength of river inputs, the local tidal patterns, the shape of the coastline, all these factors will influence how pronounced these diurnal variations are at any given beach. It's very site-dependent.
You mentioned needing nighttime data to really pin this down.
Yeah. Ideally, if you could get reliable FIO measurements throughout the night as well as the day, you could potentially separate the influences better. Tides have roughly 2 cycles per day, while sunlight has 1. Nighttime data could help confirm the role of tides versus purely light-driven decay.
But that's hard to get.
Very difficult logistically and often expensive to conduct that kind of intensive nighttime boat sampling safely and effectively.
Understandable. So why is understanding all this, the daily changes, the sunlight effects, why is it so important practically?
Well, if we want to make accurate predictions for things like daily bathing water quality forecasts or issue timely warnings.
We need models that capture these short-term changes.
Exactly. Many existing models, whether they're complex mechanistic ones or simpler data-driven ones, often struggle to capture these rapid diurnal variations, or they might rely on data collected less frequently, like weekly or fortnightly samples used for official classifications.
Right, the standard monitoring isn't usually that frequent.
No. There has been progress in developing short-term prediction models, often statistical ones, using more frequent data where available. But this research highlights the need to better understand the underlying processes driving those changes, especially these nonlinear interactions between sources, tides, and decay.
So this kind of study helps interpret what those data-driven models are actually showing.
It can, yes. Getting a clearer mechanistic insight into why FIO levels fluctuate daily is crucial for assessing the real impact on human activities, on the ecosystem, and for building more robust predictive tools for management.
Okay, so to wrap things up then, what are the key messages we should really take away from this deep dive into FIO decay modeling?
I think there are 3 main points. First, the widely used Stapleton model, while valuable, has definite limitations, especially in very low or very high sunlight, and it ignores salinity.
Right.
Second, researchers have developed enhanced models like ClipStap and RevStap to address these issues. RevStap in particular shows promise by incorporating dark decay and salinity effects and handling high irradiation more realistically.
But it wasn't always the best fit overall.
Correct. Which leads to the third point. The Swansea Bay case study revealed a real complexity. While RevStap fixed specific flaws, overall prediction depends heavily on accurately modeling the hydrodynamics, the tides and river inputs, which seem to be major drivers of the daily FIO variations observed at beaches interacting with the decay processes.
So it sounds like we've definitely made progress in refining these decay models, but predicting coastal water quality accurately is still a major challenge. There's still a fair bit to learn.
Oh, absolutely. While we've advanced our understanding of FIO decay, particularly with models like RevStap addressing known issues, there's much more to uncover.
Yeah.
Especially nailing down that precise relationship between sunlight intensity and decay rate across the full spectrum.
And needing more data.
And always needing more comprehensive, high-frequency field data from diverse locations to really test, validate, and drive further improvements in both the decay models and the hydrodynamic models they couple with.
It really does make you think, doesn't it, about how we monitor and manage these vital coastal waters, especially, with climate change potentially bringing more extreme weather and increasing pressures on our coastlines from development and population growth. How might a deeper, more dynamic understanding of these processes, the river pulses, the tidal flushing, the sunlight effects, how could that lead to genuinely more effective strategies? Strategies to really protect public health, and also these incredibly valuable and often fragile coastal ecosystems.
That's the critical question, isn't it?
Definitely something for all of us to consider. If you want to explore this fascinating area further, We really encourage you to delve into the cited research we discussed and check out the ongoing work being done by scientists in this vital field of environmental science.